The next lecture will look at a fascinating hypothesis, the pay before pumping hypothesis, actually which has been formulated in relation to the costs of digestion, as you will see in a second. What you have in this picture is basically sort of a CT scan, if you want fake in colors. This is a snake, the mouth of the snake, this is a rat basically as the snake is eating. This is something we collected quite a few years ago in Colmore, they're doing experiments with a course. Essentially, the idea here is that we are going to look following Krog's dictum. For any animal or for any process that we want to study, it's relevant to look at a particular animal that does that in a sort of a enlarged manner. Python snakes can eat 50% of their body mass in a single meal, even more. That's what you see here as a python snake eating a gazelle. And as a result of this, a couple of authors, Stephen Seeker and Jared Diamond, actually formulated a few years ago the pay before pumping hypothesis that I will talk about in a second. In connection with that, they basically published a paper called A Vertebrate Model of Extreme Physiological Regulation, relating to exactly the responses of Burmese python snakes when they eat such large meals. Burmese pythons are sit and wait foraging snakes. They have long periods between meals. They can basically fast for 1-2 months, even longer, even for half a year, and consume then huge meals, large meals afterwards. 50 to even 160% of their own body mass there are records for. So the idea is that digestive plasticity, the capability of the gut to undergo changes associated with fasting, will be largest in Burmese pythons. The experiments that are done in this respect are always along the same lines. Basically, animals are fasted for a period of time, say in this case 30 days, and then they are fed with rats, and then you take samples after several hours, all the way up to 30 days, for example, looking at the digestion phase, looking at post-absorption, trying to quantify SDA by measuring metabolic rate. The results unequivocally give you a specific dynamic action response. This is a typical SDA response, and the more the animal feeds, the larger the SDA response is. Up to 100% would be like this. What is notorious in these python snakes, and what was puzzling at the time of publication, is that you can actually increase metabolism as much, or that much. The increase in metabolic rate that has been measured in pythons from this close to zero up to the peak is 44 times. These animals can increase metabolism 44 times. This is almost unbelievable. That's what we call factorioscope. It's factorioscope. In relation with this, the authors actually started monitoring lots of other functional parameters in the gut, as we will see in a second. And in this case, you have basically the passage of food through the gastrointestinal tract. In the stomach, the food disappears after a period of 5 days, roughly. In the small intestine, you have a slightly different curve, and basically by the same time. When you look at the changes in transporters, nutrient transporters in the gut, these are the changes you see. For L-aspartate, L-leucine, two different amino acids, other amino acids here. For glucose, what you see is a gigantic upregulation of the transporters. These are transporters that sit in the apical site of the enterocytes and are responsible for, once these amino acids are digested, they are taken into the enterocyte and moved into the circulation. But notice the remarkable changes during a period of 10 to 15 days in these pythons, in the glucose transporter, but also in some of these amino acid transporters. As we said earlier in the previous lecture, this metabolic response, this SDA that we are measuring, comprises pre-absorptive and post-absorptive mechanisms. The pre-absorptive mechanisms, same lines of what I said earlier, energetic cost of gastric, pancreatic, biliary, and intestinal secretion. That is, the formation of the enzymes and substances that have to be secreted into the gut to carry the digestion. The costs of transporter and enzyme upregulation. Notice, and this is an important detail, is that for the nutrients to be able to be absorbed, they have to be there. And that has to be taking place. This upregulation, this increase in the number of transporters has to take place by the time the animal has eaten, but before actually these nutrients are absorbed. And finally, the costs of growth of the intestines and even other tissues. The post-absorptive mechanisms has to do with the biochemical transformations involved in intermediary metabolism. The nutrients are taken into the blood. What happens afterwards? What do we do with this? Glucose. If it's not going to be used, glucose is stored in the form of glycogen. Same thing for lipids, fatty acids. The cost of digestion seems to be relatively higher in the Burmese python. And because of this is related with the SDA that we saw earlier. The SDA that I showed you, basically, that it shoot up even 44 times larger. So that would appear that this cost of digestion is high in the Burmese python. And then the possibility is that this is because the tissue growth and the upregulation that has to happen before this food can be processed are higher. So the idea here is the following. An animal that fasts for a long time will atrophy the gut much more. Therefore, at the time of need, when you have to absorb, you have to put up this cost. This is basically a comparison from the Sicker and Diamond paper in Nature between the factorial increases associated to meals in pythons versus mammals. And actually they look at lots of different factors, and I want to focus on a few of them. Metabolic rate. What they say is that metabolism in mammals associated with the SDA can increase 1.5 times. Instead, in the snakes, in the Burmese pythons, they measure up to 44-fold increase. When it comes to the intestinal amino acid and glucose transports, the same thing, 1.7 to 2 factorial change. In the python, 10, 41. So what these authors are stating is that their studies show that the Burmese python do it all in big scale. SDA is a big thing in pythons. Everything increases so much more than it does in mammals. Presumably because these animals are fasting for a much longer period of time. Having said this, of course, when a group of researchers shows some interesting data in a study, there's always someone else that's going to repeat it. And actually these experiments have been repeated. The group of Tobias Wang in Aarhus, this is the same Tobias Wang that was involved in the giraffe project, actually has measured a specific dynamic action in response to feeding. 20% feeding, 20% of body mass feeding in Burmese pythons. And notice that they do see a specific dynamic action. There is an increase from this level up to 4. So basically it's a factor of 2 to 3. Not 44, but 2 to 3. If we compare now this response for VO2 with the response from the Sicker and Diamond, you see that basically this red bar is corresponding here in the graph. There are two differences here. The first one is that Sicker and Diamond start very, very low, start almost down here, close to 0. So of course when you start close to 0, the factorial change is always going to be larger. The second one is that the peak is way over the scale here. It would be up to the roof now. So, having said this, a specific dynamic action is present, but not everyone actually measures the same. In this case, it appears that this is depending on the length of fasting. Sicker and Diamond fasted their animals for 6 months. I think that in this study, the animals were fasted for about 1 month, 4 weeks. But Nevertheless, the results that Sicker and Diamond actually produced led them to define what they call the pay before pumping hypothesis. And the pay before pumping hypothesis states that most energy associated with a specific dynamic action is needed in the pre-absorptive phase. Remember, pre-absorptive, absorptive, post-absorptive phase? What Sicker and Diamond are saying is that the major cause for digestion comes from the pre-absorptive phase, when the animal, interestingly enough, cannot benefit from the energy in the ingested food. This is a crucial point in the pay before pumping hypothesis. An animal eats, but perhaps the cost of eating or digesting, the cost of digestion, is something that the animal cannot afford. The animal needs to incur in the cost of pre-absorption before benefiting from the food, the food intake. And they call this pay before pumping. The snake has to pay, there is a cost, before it can benefit from the meal. And this actually pay before pumping goes along the idea that in American gasoline stations, years ago, or say the beginning, Americans were not used to pay before pumping. Went to the gas station, put petrol, and then paid. They got the petrol in the car and then paid. But, of course, in some states, they started with the pay first, pay before pumping. And that's how actually they came up with the pay before pumping hypothesis. The idea, again, of the pay before pumping, and this is important, it has to do with the fact that it takes a cost before you can digest. So, if we want to put this to the extreme interpretation, one could say that if a python has fasted for long enough, and it cannot pay the price to upregulate the gut, their hypothesis, the pay before pumping hypothesis, would say that this animal will die with food in the stomach, because it will not manage to absorb this food, because it cannot put up the cost for upregulating the gut. That's what the pay before pumping hypothesis states. Thank you for listening, and now I'll take